Let's talk about cellular respiration a little bit more with a particular emphasis on glycolysis. So cellular respiration is traditionally divided into three different stages: glycolysis, the Krebs cycle, and chemiosmosis, and oxidative phosphorylation. Chemiosmosis and oxidative phosphorylation are within the same stage. The Krebs cycle is the exact same thing as the citric acid cycle, which we'll talk about a little bit here. So if you hear those two terms, they are synonymous with each other. Glycolysis is the first stage in cellular respiration. For cellular respiration to occur, glycolysis needs to occur. And this image right here is showing glycolysis. On the left, we have glucose, which is going to be oxidized into two molecules of pyruvate. And those are the two molecules you see on the right here. Here's one pyruvate molecule. Here's another pyruvate molecule. During the process of glycolysis, 2 ATP are produced and 2 NADH are produced. And we're going to go over that in a little more detail. To be sure, any of the macromolecules can be involved in these reduction, oxidation, or redox reactions to produce ATP. Glucose is the one most frequently used. And as far as the monosaccharides, fructose, galactose, and glucose, it's going to be glucose. 95% of our monosaccharides circulating through our bloodstream are glucose. And that's what's going to be used to make ATP. So let's take a closer look at glycolysis and a little bit of the Krebs cycle. Okay, so this right here represents free energy. The triangle is delta, and we have G. Delta G is free energy, or the amount of energy released upon the complete breakdown of a macromolecule, or the complete oxidation of a macromolecule. If we take glucose, which is one of our main macromolecules in the body, the free energy for glucose is 686,000 calories of energy, or 686 kilocalories of energy. That is to say, if we break down glucose in one fell swoop, the energy that is released is 686,000 calories. Keep in mind, the whole goal of cellular respiration is to produce ATP. And ATP is produced by coupling a phosphate to ADP. Adenosine diphosphate plus a phosphate gives us adenosine triphosphate. To couple that one phosphate to ADP only requires 12,000 calories of energy. Which means if we're breaking glucose down in one fell swoop, we're going to be losing a bunch of energy we could use to make other ATP molecules. Because if we're breaking it down and burning it and releasing 686,000 calories, when we only need 12,000 calories, that's a waste of a lot of energy. So the main goal of glycolysis is to break down the molecule of glucose in small pieces where there is energy that can be used and harnessed to do work. And hopefully we're not going to lose as much energy. Certainly glycolysis is not overly efficient. We are still going to lose a lot of energy in the form of heat via this process. But the 10 enzymatic steps of glycolysis ensure that we're not burning those 686 kilocalories all at one time. Okay, and I just want to review this equation again that we've talked about fairly frequently already up to this point. Glucose plus oxygen gives us CO2, water, heat, and ATP. Carbon dioxide, water, and heat are byproducts of this reaction. The combustion of glucose with oxygen is going to release energy that is used to couple the phosphate to ADP. This equation is including, like I said at the outset, glycolysis, the Krebs cycle, and chemiosmosis oxidative phosphorylation. Glycolysis happens in the cytosol of the cell outside of the mitochondrion. The Krebs cycle and the electron transport chain / oxidative phosphorylation happen within the mitochondria. So we have oxygen. Oxygen is required for the Krebs cycle and for the chemiosmosis oxidative phosphorylation stage. Oxygen is not required for glycolysis. I will repeat, oxygen is not required for glycolysis, though it does determine the fate of the glycolytic end products. And the glycolytic end products, as we suggested, are 2 pyruvate molecules. So the fate of pyruvate is going to be determined on the presence or absence of oxygen. Okay, so here's glycolysis. Glycolysis is a 10-step process. Now, if I put my reading glasses on, I can't read anything that's going on in this image, and I doubt you can. Maybe you can. Maybe you have much younger eyes than I do. But it is a 10-step process. Step 1, step 2, step 3, step 4 and 5 right here, step 6, step 7, step 8, step 9, and step 10. I don't need you to be able to read what's going on in this image. But what I want you to notice, in step 4, we have a splitting of a molecule. So this molecule right here is fructose 1,6-biphosphate. Don't worry about that. We'll talk about that in a minute. That's going to create 2 new molecules. The exact molecule. Don't worry about what it is. So step 4 produces 2 molecules, each of which are going to proceed downstream to step 6, step 7, step 8, step 9, and step 10. The big take-home message right here is there's 2 molecules entering step 6. There's going to be 2 molecules entering step 7. All of these subsequent steps, subsequent to step 5, are going to happen twice. Step 6 is going to happen twice. Step 7 is going to happen twice, so on and so forth. Every phase after step 5 happens twice. Okay, so we're going to look at just 4 steps of glycolysis. Like I suggested, glycolysis is a 10-step process, but we are just going to look at 4 of the steps. The very first step is the conversion of glucose to glucose 6-phosphate. We are adding a phosphate to glucose, making it glucose 6-phosphate. To do that, ATP is broken down into ADP, and certainly a phosphate which is added to glucose. So this is significant for 2 reasons. One, we are breaking ATP down, which may be confusing at the outset here because I thought the goal here was to make ATP, and it is. But the very first step of glycolysis requires the breakdown of 1 molecule of ATP. This happens in a lot of cells in the body. That is to say, the conversion of glucose to glucose 6-phosphate, because of glucose upon entering cells is immediately converted to G6P, glucose 6-phosphate, then it maintains glucose's chemical gradient, favoring it to move into cells. As long as glucose is low, glucose keeps moving into most body cells. And this is achieved by immediately converting glucose to glucose 6-phosphate. If it wasn't immediately converted to glucose 6-phosphate, the concentration of glucose would continually rise. So the conversion of glucose to glucose 6-phosphate in many cells in the body help maintain glucose's chemical gradient. Step 1 of glycolysis is the conversion of glucose to glucose 6-phosphate utilizing 1 ATP molecule. Step 3 is utilizing another ATP molecule. And I'm not telling you what the substrate is that starts this reaction because it's not important for our purposes. What is going to be produced is fructose 1,6-biphosphate. And the 1,6 is suggesting, as is the bi, that this new molecule, fructose 1,6 biphosphate, has 2 phosphates on it. Keep in mind, glucose 6-phosphate had 1 phosphate. This one now has 2, which means we have to break down another ATP molecule to add that phosphate here. So at this point, up through step 3, glycolysis has used 2 ATP molecules and no ATP molecules have been produced. Okay, let's take a look at step 7. Now, one thing you should be telling yourself right now, at step 7, this step is going to happen twice. Because as we outlined, step 6, step 7, step 8, step 9, and step 10 of glycolysis happens twice due to that split of the molecule in step 4 and 5. So in step 7, 1,3-biphosphoglycerate is converted to 3-phosphoglycerate. That is to say, it's giving up, biophosphoglycerate is giving up one of its phosphates and adding it to ADP. The breakdown of 1,3-biphosphoglycerate, the enzymatic breakdown of biphosphoglycerate, and I should add that all of these steps are aided by enzyme. These are all enzymatic reactions. The enzymatic breakdown of 1,3-biphosphoglycerate is going to add or put a phosphate with ADP to make ATP. But keep in mind, there are going to be 2 ATP molecules resulting in step 7 because step 7 is happening twice. Step 10, which is the last step of glycolysis, takes phosphoenol pyruvate and converts it to pyruvate. Pyruvate is the end product of glycolysis. And this step produces ATP. Once again, at the beginning, we suggested glycolysis starts with 1 molecule of glucose and ends with 2 molecules of pyruvate. Glucose has 6 carbons. Each pyruvate has 3. Since there's 2 pyruvate, 2 times 3 is 6. That accounts for all of the carbon that were initially in glucose. How many ATP molecules are produced in step 10 of glycolysis? 2, because step 10 is happening twice. Okay, what happens next? Right at this point, we have produced 2 molecules of pyruvate. What happens next depends on whether or not oxygen is present. If oxygen is present, the 2 pyruvate molecules are going to enter the mitochondria. The mitochondria of the cell is where the bulk of ATP is produced. If oxygen is not present, the pyruvate proceeds through the process of lactic acid fermentation, which we mentioned in a previous video, which can produce hydrogen and ions to decrease the pH of the body. But we're not focusing on that right now. What we're focusing on is what happens to pyruvate. It's either going to move on to lactic acid fermentation in the absence of oxygen, or in the presence of oxygen, it's going to enter the Krebs cycle. So I need to qualify a statement I just made. Pyruvate does not enter the Krebs cycle. Pyruvate enters the mitochondria. Mitochondria is plural, mitochondrion is singular. Pyruvate is converted in the mitochondria before the Krebs cycle. Keep in mind, glycolysis happens outside the mitochondria. So as those 2 pyruvate molecules enter the mitochondria, they are immediately converted to acetyl coenzyme A, which we see on the right here. That process is going to give off 2 molecules of CO2. Keep in mind, it's 2 because we had 2 pyruvate molecules. And it's also going to produce 2 NADH. So the acetyl coenzyme A is going to give up the acetyl group, and that is going to enter the Krebs cycle. Okay, so here's the Krebs cycle, otherwise known as the citric acid cycle. You do not need to memorize this, but a few things I want you to focus on. We see this acetyl coenzyme A right here, and the acetyl group is going to be converted to citrate, and then we begin this whole cycle. But I want you to notice 1 CO2, 2 CO2. Keep in mind, there was another CO2 produced up here in the conversion of pyruvate to acetyl coenzyme A. That 1 CO2 plus this CO2 plus this CO2 makes 3 CO2. But we're timesing everything by 2 because we had 2 pyruvate molecules, which means from 1 molecule of glucose in the presence of oxygen, we are going to produce 6 CO2, and that's exactly what we have in the equation we've been talking about. Glucose plus oxygen is going to give us 6 CO2, water, heat, and ATP. So that accounts for the 6 CO2. There's NADH producing glycolysis, which we talked about in the previous video, but there's also NADH produced here. 1, 2, and 3. Keep in mind, we're multiplying that by 2 because this cycle is happening twice due to the entrance of 2 acetyl groups into the Krebs cycle. So from 1 glucose molecule, the Krebs cycle is going to produce 6 NADH molecules. The citric acid cycle is also producing 1 FADH2 molecule. Keep in mind, NADH and FADH2 are the electron transporters we talked about. But since this cycle is happening twice, there are going to be 2 FADH2 molecules produced. So a review of the 2 processes, glycolysis and the Krebs cycle. Glycolysis produces 2 pyruvate, 2 net ATP. And I say 2 net ATP because it produced 4 total ATP. In step 7 and step 10, each of those steps produced an ATP molecule. Because step 7 and step 10 happened twice, that's a total of 4 ATP molecules. Keep in mind, in glycolysis, step 1 and step 3 hydrolyzer broke down an ATP molecule. That's 2 ATP molecules used, 4 produced, so we have 2 net ATP at the end of glycolysis. We also have 2 NADH produced. NADH is the electron transporter or shuttle that shuttles electrons to the electron transport chain. The Krebs cycle produces 6 NADH molecules and 2 FADH2 molecules. So from glycolysis in the Krebs cycle, how many total NADH molecules are we producing? In addition to that, the Krebs cycle is producing 2 ATP molecules and, of course, so 6 CO2. And I need to qualify 4 of the CO2 are actually directly from the Krebs cycle. 2 of the CO2 molecules happen in the conversion of pyruvate to acetyl coenzyme A. So one thing I want you to notice is after glycolysis and the Krebs cycle, we've only produced a net 4 ATP, which is not very much. So I will argue all day long that the single most important aspect of glycolysis and the Krebs cycle is the production of NADH and FADH2. The 8 NADH molecules and the 2 FADH2 molecules, which are going to shuttle electrons to the electron transport chain in the inner mitochondrial membrane, which is going to end up producing upwards of 34 ATP molecules, which is a lot more than these 4. That's why I suggest it's the 8 NADH and 2 FADH2 that are much more important products of glycolysis and the Krebs cycle. And that is glycolysis and a little bit of the Krebs cycle in a nutshell. We'll talk more about chemiosmosis and oxidative phosphorylation